Welcome to the Explore Global Health podcast. I'm Dr. Rob Murphy, Executive Director of the Havey Institute for Global Health here at Northwestern University Feinberg School of Medicine. There are many hurdles to diagnosing infectious diseases in low and middle income countries. A major problem is the lack of laboratories with proper equipment and trained staff who can get test results back to a patient quickly. Technology developed by Northwestern University engineers is about to change that reality. A new, highly sensitive, easy-to-use test for COVID-19 has received EUA, or Emergency Use Authorization status, from the U.S. Food and Drug Administration, the FDA. This technology platform, called DASH, stands for Diagnostic Analyzer for Specific Hybridization, hence we call it DASH, requires just a nasal swab swipe, and 15 minutes later, it delivers a result with the accuracy of PCR technology. The plan is to roll this technology out for COVID testing now and soon to diagnose other diseases and infections, such as sexually transmitted diseases and any other infectious disease in people anywhere in the world, but particularly in low and middle income countries. Sally McFall, co-developer of DASH, is here with details of this homegrown technology. Dr. McFall is a research professor of biomedical engineering at Northwestern Engineering and director of the Research Center for Innovation in Global Health Technologies, which is based partially at the Havey Institute for Global Health. She has extensive experience in technology development in low and middle income countries. Sally, welcome.
Thank you, Rob.
Back in 2017, you and Dave Kelso, clinical professor of biomedical engineering here at Northwestern, developed DASH and a spin-off company called Minute Molecular Diagnostics for this new technology. Let's go back to 2017 and tell us how this all began.
Dave Kelso and I had some ideas about how to make a point-of-care PCR test be completed in the time that it takes for the patient to be at the doctor's office, for example. So we had read some different reports, particularly from Charlotte Gatos's group, that people would wait for 15 minutes for a test result. But if it got longer than that, they just weren't able to wait. They had things they had to do, and they had to move on. And so that was our goal, is to make our test 15 minutes or less. And so we started working on the technology together.
And so Charlotte Gatos, by the way, is a professor at Johns Hopkins University that you were collaborating with at the time, correct?
Right. So I had received a grant to develop technology from the Johns Hopkins Center for Point-of-Care Technology Development for Sexually Transmitted Diseases. And so that was one of our first tests that we were developing for DASH. Our model assay was for chlamydia diagnosis. And we were also developing tests for other specimen types. So for a test to work, you need to be able to extract the DNA or RNA from the sample and then amplify it and detect it with PCR. And different samples can be more challenging for different reasons. And we want our platform to be able to use all different types of specimens. So when we developed the test, we had to test it with a lot of different specimens. So we tested with vaginal swabs, with nasopharyngeal swabs, and with whole blood.
Well, that's great. We're still working with Charlotte and colleagues at Hopkins in the whole POCR network, this point-of-care technologies research network. That's an NIH-sponsored entity that's really taken us all a long way. You want to tell us exactly how being in that network has helped the development process for DASH?
Well, sure. So Rob and I are the co-PIs of one of the centers that in this POCR network, our Point of Care Technology Research Network from NIH. So when COVID hit, NIH requested that POCR pivot to addressing COVID-19. So I happened to be kind of at the right place at the right time because, I also had been thinking about pivoting to test COVID. Back then, it's kind of funny. I thought, maybe it won't last that long and I won't have time to develop a test and stuff like that. But I decided to develop a test for COVID, and I was able to work on that and developing both the sample prep and the amplification. And I had even thought, well, maybe I'll just use it to test the lab to see if we're all safe, during this kind of bad time. But anyway, so then the POCR network was tasked by the NIH to lead the RADx program, the government's response to developing diagnostics for COVID-19. And so Dave and we applied to RADx, and at the time our technology wasn't mature enough. And so they did what they called diverted us to other funding source. And so we were actually funded by the POCR network and a grant from our center. And that money enabled us to develop to the point where we were fundable by RADx. So we kind of, we caught up.
The NIH poured all this money into this RADx, Rapid Acceleration for Diagnostics for Coronavirus. How fast did that speed up your development? Did it speed it up by 1 year, 2 years, 5 years?
I'd say 2 years.
2 years.
The RADx award to Minute Molecular was $21.3 million. And that was to allow us to scale up, to take our technology and be able to produce enough tests to, make an impact in the pandemic. And I don't actually remember exactly how much the award was for.
$3.7 million.
Okay. $3.7 million through the POCR network through CTHAN that allowed us to kind of catch up and get to the place where we could be eligible for that RADx award.
Yeah, the only reason I bring it up is that, everyone always says, oh, the government is so slow and, everything. I mean, this started, day one was April 29th, 2020. And it really is amazing what has come out of that. And one of the star developments in that whole program is the DASH project, to get a 15-minute PCR developed at a spinoff from a university. It's really been incredible. Let's go on and talk about some of the other issues related to that and to find out a little bit more about you, because you're just such a fascinating person who's developed this incredible 15-minute PCR device. What drew you into wanting to improve point-of-care testing for infectious diseases such as, let's go back to HIV, TB, and hepatitis, and specifically for low and middle income countries?
I've always wanted to have my work have impact. I'm a scientist, so I wanted to approach it through a scientific method. I'm not the kind of person that's going to go out and be like on a board for a charity and raise lots of money. I'm more introverted than that.
You can always do that,
I decided I would just try to use what skills I have, the talents I have to try to make impact. When I found out about molecular diagnostics and started working with Dave Kelso on point of care technology, it just makes a lot of sense to me because, the patient can receive the result in that same appointment. And if there's a medication that's required or even just the communication to the patient, this is what we're going to do. It makes so much sense. There's, so much less loss to follow up. And if it's an infectious disease, you can cut down on transmission. So I just think that it makes a ton of sense for this kind of approach in our, higher resource countries, but even more so in low and middle income countries.
But it has applicability here too, as well. Imagine to go into one of these urgent care centers and want to have a test for COVID or whatever it is, and to have them take one of your cartridges and stick it in the machine after they get the specimen, whatever the specimen is, and get a PCR answer in 15 minutes. It is really very dramatic.
Yeah, we're really excited about that because we think we can make a difference in things just like transmission, right? So you can help someone improve their health by, especially with these medications now for COVID, that, the antivirals, you can shorten their infection, help prevent them from spreading it to someone.
That's a good point because, now it's all about test and treat. The two drugs that are approved right now are for early treatment. You got to start it within 5 days of your symptoms. And if you don't have any symptoms, 5 days of your test. And a lot of people, by the time they even get the results back and all that, they're past the 5 days. So timing is really critical.
Especially in our population where we have people who are vaccinated and when they become infected, their immune system is working. They get symptoms maybe even earlier than they would have if they hadn't been vaccinated. But they're not antigen test positive. They're not home test positive. And so if they have symptoms and they're able to get a PCR result like with DASH, then they would be able to immediately be given the antivirals. So especially with, in high risk patients, I think that's really critical.
Compared to the tests that are out there right now, the ones you go into Walgreens or CVS and you buy a test for COVID, let's just start there, but it could be for anything else. We sell a lot of different tests there now. Besides the speed, those tests are also about 15 minutes. Besides the speed, what's the difference?
Well, that's exactly what we were addressing, is that you can get a test, that kind of test that you can buy at the drugstore, and the result will be there in 15 minutes. But it is not as sensitive as PCR. A PCR test can be positive about 2 days earlier than an antigen test. So if you have symptoms, you go to the store, you get that test, you test yourself, it's negative, then you're supposed to wait a couple of days and then test again. And that's great if you are able to do that, if you're able to stay at home and isolate, and, but if you are not able to, a test like ours, if you know that you're positive, then you can alert your employer. You can kind of activate those systems where people understand why you have to be at home, rather than just you say, I may or may not have COVID. So yeah, the test is a lot more sensitive, and it's also easier actually to run, because this swab just goes right into the cartridge. You break it open, you break this swab at this break point, you cap the cartridge, put it into the instrument, and the result is in 15 minutes.
Well, speaking from experience, just recovering from my 3-week COVID experience, I can tell you it was just a sore throat and laryngitis. And a little fever. Now, so you developed this technology thinking of low and middle income countries in mind. Why is this kind of technology needed in a low and middle income setting?
So there are a number of things. One is kind of logistics. In our healthcare system, if you get a test at your doctor's office, they might send the sample to a testing company, and then the result is returned to the doctor. The nurse would let you know, or maybe it's on you log in and you can message your doctor, and you might see your result there. But in a lot of low and middle income countries, they don't have all that infrastructure. Once they collect the sample, if they send it to a lab, it may take a few days for the sample to get there.
We did one study with Carapal Mountain looking at doing sophisticated testing in a setting such as that in a low-income country. And not only did it take days to get the result back, this was regarding infant HIV diagnosis, but the study that she presented, it was, I think, 40% of the people never came back. And that's true everywhere. Everyone doesn't always come back for their test results.
Or if they come back and they don't get the result, then they feel disenfranchised, right? And then they tell their friends, don't even bother.
And plus the lab is paid for that already. PCR typically is expensive, relatively expensive. So this could be a real backbone of some laboratory, particularly in a place where there's not access to big central labs or an emergency room or someplace like that. How effective is it? Can you put some numbers on this?
So in our clinical study that we used to apply to the FDA for emergency use, our sensitivity against another emergency, an EUA approved test was a little bit above 95, so like 95.5% sensitive, and our specificity was, I think, 98.5%. So we're highly sensitive and specific to this other test that is more laboratory based.
That's great. You mentioned a couple sample types. What sample types can you put in there? Can you use saliva? Can you use nasal swabs?
Yeah, definitely. So we use nasal swabs. So a nasal swab is what we received our emergency use authorization as a specimen type, but we've also tested saliva, and that works. We can use swabs that go into what's called viral transport medium, which is that pink liquid that a lot of hospitals like to use that. So if they took a sample from a patient, they could either use DASH, or, if they had a different test they also wanted to use the sample for, they could use that.
So it doesn't really make any difference. You can use kind of anything.
Yeah, anything for COVID. We could use a throat swab. And then looking forward, our next test is going to be a test for flu and COVID, so that when a patient comes in and they have respiratory symptoms that kind of overlap, is it flu or is it COVID, we'll be able to distinguish that.
And that would be in the same cartridge?
Mm-hmm, in the same cartridge.
In the same test. So you just put the one swab in and it'll come back both negative or positive negative?
Either flu type or SARS-CoV-2
Wow.
COVID. And then we're also developing a test for strep throat because, again, what we were thinking is if you have our analyzer, what else would you like to test for? So if you're testing for COVID, you'll probably want to test for other respiratory infections. And since especially Omicron starts with a sore throat, we thought Strep A would be a good thing to detect.
Now, it takes 15 minutes, but you can only do one of these at a time, right?
That's right. If you need to, have a higher throughput than that, then you could have more than one DASH analyzer, and they can be linked together. So you can have one kind of teacher and the other pupils, or however you want to think about that, so that one administrator could kind of manage all of them.
Tell us a little bit more about the equipment, the DASH. What does it look like? How big is it? Is it heavy? Can you carry it around?
Yeah, it's about 3 to 5 pounds, I think. And it looks a bit like it's about the same size as a box of cereal. It's a really pretty blue. It has a clock in the front with lights that kind of go around in a circle and show you if the test is nearly done. So that kind of helps you if you're put the swab in and now you're over talking to another patient, you can kind of look over and see if the result is going to come out soon.
So there are definitely machines that you're using. You have 10 in your own lab. Where were those made?
So those were made, we have a consulting firm. They've done a lot of the hardware and software development. And we also have a mechanical engineer we've worked with for many years, Tom Westberg, and they built those analyzers.
Are they here in Chicago or?
They are. It's the DeMonte Group. And then they taught our contract manufacturer how to build them.
Oh, that's great. Wow. So really, truly a local development project.
It is. It's totally local. Yeah, we've all worked together for many years. So this has been really nice culmination of all of our collaboration over the years.
So when somebody purchases one of these, the cartridges, how do you go about training the user?
So for our emergency use testing, that's a requirement from the FDA is that you just give them you give the users a pamphlet and they're supposed to be able to run the test without any training. And that was successful. But when we do supply the analyzer and the cartridges to a customer, I think we want to give them a little bit more than that. We'll come and make sure that they're comfortable.
How sophisticated do you have to be to actually operate the machine?
So, our authorization is for a place that would have a CLIA license. But as far as being able to run the test, you don't need very much sophistication at all. You just need to be able to break the swab, put it into the analyzer.
Like who could do it? Could a student do it? Could a administrator do it?
Definitely. In our emergency use authorization clinical study, the people who did the tests were not laboratorians. They were study personnel, nurses. I think there were some students.
But you don't even have to have a medical background.
You don't. No, that's true.
So now that you have this EUA approval, emergency use authorization from the FDA, what's happening like right now? Are you selling any of these things or marketing them, or what are the next steps?
So our rollout plan is to begin to sell our analyzers and cartridges kind of midsummer time. Right now, we're scaling up the production of the analyzers and the cartridges. So we're working with contract manufacturers. We're transferring the technology. We're going through all the kind of documentation that we need to have in place to sell a regulated product like this.
The EUA is for COVID, right?
Yes.
Okay. Now, originally, this whole machine, this technology was developed for HIV viral load, other infectious diseases like influenza, hepatitis C, as well as sexually transmitted infections like chlamydia and gonorrhea. How soon are you going to expand beyond coronavirus testing?
That's going to be as rapidly as we can, because we think that's the real strength of DASH is it's a platform. It can be used in a number of different tests. And in fact, we have a plan to expand to a menu that has 26 different analytes or targets.
26 targets?
Yeah, not all at once. But 26 different tests that we'll be developing over time.
You're going to actually have to have a catalog. Besides what you've mentioned, what other things are you looking at?
So there's flu A, there's flu B, RSV, sexually transmitted diseases, chlamydia, gonorrhea, TB, trypanoma, HIV, HCV, HBV. We could do other kinds of genetic testing. Anything that you want fast, I think, is something that we could do on DASH. So if you needed to test for relative drug sensitivity, like warfarin kind of thing, and know what kind of drug to give to a patient, we could probably do something like that on DASH.
Probably one of the most common questions I get is, I'm going to a wedding or destination event, 150 people, and we want some kind of test to do for everybody. Right now, a lot of those places are testing with the rapid tests, the over-the-counter rapid tests. But like you said, their sensitivity in asymptomatics, first of all, most of them are not approved in asymptomatic people, but the sensitivity is not that good. Could DASH actually be used in some situation like that? I mean, I just picked that. That's just because I get that question a lot. But I mean, it could be a workplace situation. It could be the gridiron dinner in Washington, D.C. Maybe they would have done a little better if they had a DASH machine before that thing. But, being vaccinated obviously is not enough. Do you see anything like that?
Oh, definitely. So first of all, DASH is also not authorized for asymptomatic patients. We didn't test for that.
Do you think you'll apply for asymptomatic testing at some point?
It's such a difficult category because FDA considers somebody who has been exposed as somebody who, say, your next door neighbor tells you that she has COVID and you had been having coffee with her the day before. And you just want to know, do I have COVID? You don't have any symptoms. But when you go to be tested, you're not considered asymptomatic in the FDA's test. You have to be not suspecting at all. So you have to test a lot of people to find those few that were exposed and weren't really even aware of it. A lot of companies and physicians use a test like ours or even the rapid tests with asymptomatic patients, and they're allowed to do that. And it might make sense to use DASH in that way before a wedding or before travel. For me, I like to know that I don't have COVID or I'm not harboring the virus before I see my mom who's 90 and my dad who's 92. So I could see that type of testing, before you go to visit your relatives or before a wedding, or, a meeting.
No, that's incredible. Sally, it really is just incredible what you and Dave and Kara have done with Minute Molecular Diagnostics. I hope people appreciate that full-blown thermocyclic PCR, which is like the best, is now available for COVID and simple to use. I mean, it truly is amazing. My last question to you is, what has it been like to be part of creating such an important technology, a real potential game changer like this, that has the potential to impact so many people around the world?
I just feel really proud to belong to this team and just the work that the members of our research team have done to combine their different skills, their different experiences into something that, really works. So when we first had the idea of a 15-minute test, we didn't know if we would be able to do it or not. It was kind of pushing the envelope. That's what some of the lab members said to me when we got the EUA. They were like, we really actually did it. It's like we just kind of just realized it, but it's been incredible to have this opportunity. And also just with the POCR network, they've been so supportive. The POCR network is in their third cycle, and our lab has gotten funding from each of the cycles. So we're really kind of the baby. And we've just kind of developed and matured along with it. I'm really proud of all the hard work that our team has done to bring this all together. It was, we've had software engineers, hardware engineers, biologists, chemists, all working together to create this. And within the cartridge, everything kind of flows from the sample to the answer. It's not like the chemist did this and the biologist did that. Everything has to fit together, and that's how we work together as a team to do it. Also, just working with you, Rob, all the way through COVID, both of us on that RADx program. You're being on our advisory committee for the CTHAN award and kind of helping guide us through that period of time as we tried to catch up with the RADx. It's just been really rewarding.
It truly has. I was just proud just to be part of it. I was really a very peripheral person in the whole thing. But it's amazing. And again, I already said it once, but I'm going to say it again. I mean, everyone always is complaining the government can't do anything. And, look at what's happened during COVID. We are in a pandemic. We developed vaccines to the market within 10 months. And we've revolutionized testing. I mean, there are billions of tests now available. And now a 15-minute PCR test. I mean, it really is incredible. It reminds me a little bit about PEPFAR. That was the President's Emergency Plan for AIDS Relief, when basically most people had just written off Africa and many low-income countries, just saying, well, they're never going to be able to afford it. They're never going to be able to distribute it. They're never going to be able to monitor it. They're never going to, never, all this, no. And PEPFAR was a huge success. And this is kind of in the same genre as that, people said you couldn't do it. And then it gets done, and it's great, and it's a game changer. And it's been so fun working with you, and it's so fun talking to you today about your experiences over the last couple of years and getting this product out there, our homegrown Northwestern and Chicago product. And I'm sure everybody that's listening to this is going to be getting something tested at one of these times. So it's really quite amazing. So thanks for joining us, and thanks for all the work. I'm so proud of you and this entire project. Thanks again.
Oh, thank you so much, Rob. Thanks for having me on your podcast.
Follow us on Apple Podcasts or wherever you listen to podcasts to hear the latest episodes, and join our community that is dedicated to making a lasting positive impact on global health.